Many people know Jeff Milton, right? Jeff is not just the founder of La Jolla Labs, but also run it as a CEO. And if I saw the news correctly, now you're switching with your co-founder and focus on technology, and she does the CEO and has to deal with all the HR issues and so on. So I think it's a relief, is it? So for those that don't know La Jolla Labs, and he will talk more about it, a company that also thinks about RNA therapeutics and modalities. So, but it's not the first time he does it, right? So you have been working with, as a head of data science with Arcturus Therapeutics. You have been also working, of course, as a director of functional genomics and drug discovery at Ionis, right? And then before that, you also had time with Genentech. And so we know you, right? We work with you. Neon Way has worked with you before, and we're looking forward. What are the news? And yes, so Jeff, actually, I just also have this on my list here. You are a graduate from Mellon College of Science at Carnegie Mellon University. Yes, okay. So welcome again. Glad you made it safely, and here we are.
Great. Thanks. Yeah, so it's going to be a continued talk on ASOs, but this is actually a talk I've wanted to give for a long time, and it's a work in progress, because what I wanted to actually talk about was actually more about the modalities that are used. One of the things that I experience a lot at La Jolla Labs. So a little bit of history. We started La Jolla Labs. My co-founder and I, Tamar Grossman, started La Jolla Labs about 3 years ago. We're both from Ionis Pharmaceuticals. We were at Ionis when Spinraza was made during Millicin. So there's a lot of overlap. In fact, you're going to see some slides that I have that Stan Crook gave me that were just shown in the preview. But that's fine. That's good. But in any case, there's a lot of overlap. And so I'm going to come at this from a point of view of a company. How do you run a business doing rare disease? It's very difficult. And so every time when I get into this thinking about drug discovery, I think about it from the point of view of how can we make a business out of it that breeds innovation, that hopefully kind of perpetuates and makes it sustainable. So that's kind of the angle. This particular talk is really because I've always wanted to talk about the different modalities of drug discovery and the context of rare disease, because rare disease, you have to think of it from the point of view of rare disease and not be sort of stuck with a technology trying to fit it to the disease. So this is part one, because I'm going to talk about it in the context of chronic dosing versus gene therapy. So I'm not going to talk about gene therapy, the one and done type scenario, because that would be part two. But you've seen this before, rare diseases, it's a long tail. And what that means is that from the population, or from a patient perspective, there are a lot of different individual patients that you have to focus. So you have to be more individualized. And that creates a very different landscape. The conventional approach to therapeutics development and even clinical trials is totally different. You have to interact with patients. You have to talk to families and kids, and there's no Hippocratic oath when you're in business. So you have to ride this very fine line between what's right and what's wrong, because you have to answer to the patients. Ultimately, that's what you have to do. But you also have to make a business. So that's the struggle, is how do you really do that? And when you think about the fact that 90% of drugs fail in sort of the common approaches to clinical trials, 90% fail, that doesn't work for the business that we're in. So there's a lot that has to be taken into consideration. So I get parents talk to me all the time, and they say, we want to do an ASO. And because it's a rare disease, and they've heard about it, and they think about the technology, and I say, I don't know if that's what you want to do. It's probably about $700,000 from start to finish, if you're lucky. And so what I say is, try to look at repurposing. So is there a way to repurpose a drug that's already out there? So there are companies that do this. One of them is, I'm blanking on the name, but it's Unravel. If you're familiar with, anybody familiar with Unravel? So they'll, for like $15,000, they'll do a screen for a particular disease, and then they'll find other particular indications that might be linked to it. They have a business model that somewhat works. I say, go to Unravel first. It's cheaper and probably faster. The tox profile, the preclinical work is super, super expensive. And a lot of people who do rare disease don't even think about that part of the work. They kind of do proof of concept, they get excited, and then they get into the preclinical side of things and the money runs out. And so you end up with a lot of dead-end projects. So let's talk about small molecules. So small molecules, I started this industry when rational drug design was kind of the approach. It was kind of the, Gleevec was the idea of taking a protein from the PDB and finding the pocket that you want to get it into and designing the small molecule into that little pocket and rationally design the drug to fit into the target that you want and make it do what you want it to do. But ultimately, is it really rational? There's a lot of space you're playing around with there. Maybe AI can help, but as we're finding out, there isn't enough data to really figure out, to really kind of make that happen in a super efficient way. It's still 10 years to do a drug and about $1 billion. So that's kind of the small molecule approach. Can it be selective? Well, here's one that's actually pretty selective. So this is a drug by Merck, and it interfaces or interacts with this MET kinase inhibitor, so a mesenchymal epithelial transition layer, and it's very, very selective. I think it's like 10,000-fold more selective than any other kinase. So it's very selective to that particular target. So that's still a very costly enterprise if you go down that path. So what does that look like? So for a small molecule world, this is kind of what a typical small molecule approach would look like. You'd have a big or a large library of 1 million different compounds. In this particular case, the screening library is what's called a fragment-based drug discovery. There are other types of small molecule libraries, diversity libraries, things like that, that you can do these millions of compounds of screens. And then you find 500 hits for the particular target you're looking at. First of all, the target has to be extremely well characterized. And then maybe you can do crystallography to figure out how that fits in there. And then you can do some rational drug design, some med chem to figure out, I've got to make it look more like a drug and have the properties of a drug so that it sits around and does the actual, what it's supposed to do without being cleared too quickly, that type of stuff. But again, you're still looking at, in this particular case, I think this was about, so this is under 10 years, so that's quick. So this is kind of giving you an idea of the benchmark that we're dealing with a small molecule. And then this one is actually, this will be the last one. So this is from Vertex. And this is the ETI drug for cystic fibrosis, or I'm sorry, CFTR. Yeah, so cystic fibrosis. And what's cool about this is actually, this one is actually, it binds to a lot of different, it's a folder enhancer and potentiator. So you can kind of look at the phenotype of this and screen for that. So that's kind of cool. And this was actually pretty quick. I think it was less than 10 years, but they have these nice cryo-EM structures over here that show the structure. So that's nice. So the one thing I want to say about this is that there are a lot of off-target effects with this. So there's liver tox, there's kids get cataracts with this. So that's the other problem with small molecules, is that you can get very promiscuous hits in a sense. So now we go to antibodies. So this is a separate modality. This is much bigger structure. You have the variable region, the antigen binding region, and then you have this very conserved region here. So this is the part that you're going to bind to the actual, whatever it is, the molecule you want to bind to. And then there are all these different modalities that you can chop up and use separately. So for example, there are some enzyme replacement therapies that use just this Fab fragment as a delivery vehicle. So they're very versatile, but very complex. And this is one of those structures that gets into sort of downstream costly effects with synthesis and characterizing structures. So in any case, this gets very complicated. I'm not going to go through all the details, but they're very versatile in the sense that they can be used as delivery vehicles. So in this particular case, you've got an antibody with a linker and then a payload. And so for cancer purposes, that's great because you can have the antibody bind to the tissue, the specific cancerous tissue, internalize, and then the payload is the toxic part for the tissue. And then you can have something like an antibody bound to an siRNA or an ASO. And so these are structures from Avidity. I think I took that from their website. But that shows how you can go even more selective. So you can be tissue selective and then target selective, RNA selective. So that's kind of gives you some of the idea that antibodies go a little bit further as far as selectivity than small molecules. But there is a very big downside to antibodies, and that is the cost of some of the synthesis and some of the downstream preclinical studies. So when you scale up these reactions, they can be very expensive. I mean, so I was at Genentech for many years, and I used to go into the scale-up facility and talk to some of the guys there, and they were saying, I'm about to run $1 million reaction, so don't talk to me. Because if they mess up, that's it. So in any case, there's a lot of complexity that goes into this. And when you're doing things like making or manufacturing this, you have to characterize what they look like. If it's an antibody drug conjugate, these things are actually in solution in ratios, and you have to kind of find ways to develop the assays so that you can actually characterize what those structures are, so you know what you're delivering. So that's an extremely expensive downstream enterprise, but it's very selective. And so, of course, this is kind of a dump of all the different things that you need to do for taking a drug, antibody from beginning to end, from cells, from sort of research all the way through to the IND filing. But this is the type of stuff that gets very costly that people don't see. So part of the reason why I love RNA is because it's targeting the genetic cause of the disease, and it's very simple as far as the modality itself. So for the rest of the talk, I'll talk about the RNA sort of landscape. And this kind of gives the history of it. So Ionis started in 1989. And so right about here, it was started with $7 million right here in Sorrento Valley by Stan Crook, the founder of N-Lorem. And they went through a lot of ups and downs. It was not an easy road by any means. But in any case, Stan persisted and really worked this out. And you can see that really by the number of publications. He's very, he publishes quite a bit on this. And so it's you can really kind of follow the whole path that he did for this. But the first drug was actually Formotersen. It's off market now, but this was for it was for HIV patients who had a viral infection in their eye. And so, And so yeah, so that was the first drug. And I think that was a straight DNA. I don't think that was chemically modified. It might have been chemically modified, but very little. In any case, and then we go, yeah. So anyway, I'm going to skip through some of that rest of that stuff. I don't think they have Spinraza up there, but Spinraza was 2016. And that was the first blockbuster ASO drug. So that was, and that is a steric blocking drug. I'm not going to go through the mechanism of that, but that was it wasn't a gapmer. It was basically just a molecule well, I'm getting too deep into the science, but basically it's a steric blocking ASO. So it was fully modified ASO. And so these are the two RNA targeting drug modalities that are typically they're talked about. So this is the Ionis approach, and this would be the Alnylam approach. So the siRNA versus the ASO, single-stranded ASO. And so the mechanism of action is basically, for the gapmer ASO, is it binds to the target RNA. The RNase enzyme recognizes this as basically not supposed to be there, and it cleaves it. And then you have the RNA degradation. And then the siRNA is a little bit different, but it's the same basic mechanism. And they can be viewed as catalytic in the sense that these molecules actually don't get cleaved right away, but they can go and bind to other RNA targets. So, and the big difference here is that this happens in the nucleus, whereas this is in the cytoplasm. So the siRNA doesn't actually happen in the nucleus. And so there are some, that's part of the reason why, so there's some advantages to the RNase approach when it comes to cleavage, because you can cleave in the intronic space versus the siRNA has to be on the mRNA. And so the siRNA, and in this particular case, actually this is the steric blocking approach. So for the steric blocking approach, it's the same type of molecule, but it can actually get into the nucleus. So for steric blocking, for things like exon skipping, you might have seen with DMD, this all happens in the nucleus. And so you can see that it can bind to various locations in the intronic location and cause various splicing factors that would either enhance an exon or skip out the exon. And that's the mechanism of action of Spinraza. And so here's the stand slide. So there's basically a lot of different approaches to RNA, this particular modality. And this is really the reason why we really like it. So you can do the RNase approach, which is the gapmer approach, the siRNA targeting RNA, and then nonsense mediated decay, which in a second I want to show you a little an experiment that we did that we really, one of the reasons we like the nonsense mediated decay is because we can use a fully modified ASO. It's not a gapmer, has a longer half-life in the CNS, and it's much more durable compound. So we could potentially use it systemically or in muscle. And then there's no-go decay, which is basically pausing the ribosome so that they basically, the body says, okay, this is not working out, so we'll degrade this. And then translation inhibition and alternative polyadenylation. I'm not going to get into that, but what we have done at La Jolla Labs, and then there's microRNA strategies for increasing expression. This is kind of like our, this is essentially what we're going toward, is trying to build a palette of experience for being able to do any of this work. So we've done upregulation with microRNA inhibition and enhancing canonical splicing. And don't think I'll show that one, but we have not been able to reproduce this, although it has been published at Ionis. And then we have downregulation. We've done RNase-H. Turns out that's a much easier process than our favorite, which is inducing NMD, which I will show in a second. And then we have done a pretty good allele selective knockdown, which is the n-lorem approach. And then we have done exon skipping and cryptic exon modulation. Protein engineering, I don't think I'll show that, but what we're able to do is essentially move a splice acceptor site so that we add amino acids into the active site of an enzyme. And we think that would attenuate that enzyme and essentially knock it down. So that's the way we would do protein engineering. So this is an example of an NMD. One of the reasons we started La Jolla Labs was because of a disease called Lafora. I've talked about this before, but this is a glycogen storage disease. These are kids that are high functioning into their young adolescence and then start a cognitive decline and end up dying in a vegetative state. It's a terrible, terrible disease, very rare disease. So what we have, and this is our software, our La Jolla Lab software on the top here. This is an AI model that we have that shows us how to splice out an exon, essentially. So what you see with this dot right here is, this is an exon, this line. The bars going up are attributes we call cis-regulatory attributes that are associated with the inclusion of that exon. The bars going down are for the exclusion of the exon. So we would put our ASOs, which are steric blocking ASOs, on these blue dots. And those are hitting the bars that are going up. So we want to cut the exon out. So we know where to place them. And so I think we had probably about 30 ASOs for this one because we knew where to put them. We cut out the exon. That caused a frame shift, and that induced nonsense mediated decay. And so we did this in a dose responsive way. And then we showed it with protein. And then we also did a phenotypic assay where we're actually knocking down glycogen the way we wanted to. So what we did was we created a molecule that was fully modified so that it wasn't consumed by nucleases. So we didn't want the Gapmer approach because that has a shorter half-life in the CNS. So this, theoretically, the patients would be dosed once, twice a year. And it has that durability with this particular approach. So it's the same molecule as Spinraza, just a different sequence. The same chemistry as Spinraza, different sequence. And here's an example of what that looks like. So this was really the reason we decided to go this route, was because Spinraza has such a long duration of action. So we wanted that chemistry, just a different biology. So the last part of the modality talk is really where RNA is going, which I'm kind of excited about. I mean, there are these ADA-imers, which are the RNA editing. For the first time just recently, we went into the clinic with that. Wave went into the clinic for this particular target. I think I've had probably over the course of the last year, 2 families come to me with single nucleotide mutations in acceptor sites of splicing acceptor sites, which could be done with, they could have fixed it with this type of approach. But it was so new that we're not prepared for it. We're not ready for it. But I think that's coming. So I think this is kind of an exciting thing to see. But the other thing is, does this, we're kind of waiting to see what Wave kind of reads out and how this goes. But the other thing is also, it still has the same delivery problems that every other RNA modality has. And we'll have to work through that. But in any case, they have this PRISM chemistry that they talk about that I guess makes it very durable. It is a larger molecule, so it has a sequence space that targets the actual RNA, but it also has a part of the molecule that recruits the enzymes that does the base switching from an A to an I or A to G. And so they actually show some pretty compelling data that shows that they're actually getting this in vivo. And then they have some pretty good clinical data coming out here pretty soon. And so I think there's a lot more that's going to be happening in the RNA space that I'm really excited about. And so this is what the RNA editing space looks like right now. So there's a lot of stuff in preclinical, but Wave now has that first clinical in phase 1 right now. And I think they did read out some positive data on that. But ultimately, when we're talking about rare disease and the approach, it comes down to a combination of how much money, how much time, efficacy, safety, money and time are basically the same thing. But you've got to build that therapeutic window in a short amount of time with a certain amount of money. Is if you can get a situation where you have multiple shots on goal, that's the best. If when I see these families come to me and say, we want to do an ASO, I think, that's one shot on goal, and it's probably not going to work out. It's going to take a long time to get to that point. And they're putting their own money into it. I try to kind of manage that process. Because ultimately, when you think about just the manufacturing costs, anytime you go from, I like to look at this from the point of view of chemistry and biology. If your manufacturing process involves biology, it's going to be expensive, no matter what. That's just the way it is. And in chemistry, it just has a little bit more of you can characterize the compounds a little bit better, and you can manage it a little bit better. But this is the landscape of what things look like. I did my best at kind of figuring out these numbers, but for the most part, if you can find a small molecule approach, that's the best. And then ASOs are next, and then goes from there. But this is part of the reason why you see, if you look at a lot of the companies that are really in distress, you'll see a lot of these are in the cell gene therapy area, because that late stage price tag is astronomical. And with that, thank you very much.